Today, we will describe and show step by step the technical procedure used for analysis of RNA sample by hybridization on microarray. This is an example of a technology used in the field of transcriptomics and provides a quantitative determination of all messenger RNA and a large amount of non-coding RNA present in a biological samples. Recently, other techniques based on next-generation sequencing, such as RNA sequencing, have been set up for the same purpose. However, hybridization on microarray is a well-established method which has been progressively improved in the last 2 decades and can be considered a robust and reliable methodology. The microarray technology is based on the hybridization between target nucleic acids and probe nucleic acids. We call target the nucleic acid that is the object of our analysis, and probe the known nucleic acid that is used as a tool for the analysis. In the microarray technology, millions of hybridization reactions to different probes are performed simultaneously. Probes are fixed to the surface of a solid support in a specially ordered way in a bidimensional array, while the target DNA is free in solution and can be labeled with the fluorochrome. It can be considered a form of reverse hybridization. After hybridization, the unbound target is washed away, and the position on the array of the hybridized target and its fluorescence intensity are revealed by a laser-based fluorimetric scanner. Today, we will use an array produced by Affymetrix called Human Transcriptome Array 2. In this particular array, probes of 25 nucleotides are covalently fixed to the surface of the microarray. This high-resolution array contains 6 million of different probes. 70% of these probes cover exons of coding transcripts, and the remaining 30% cover splice junction or non-coding transcript. On average, 100 probes per gene are used. Let's start now with a brief overview of the protocol.
The entire procedure can be subdivided in 4 main stages. Rna extraction, preparation and labeling of target cDNA, hybridization of target cDNA to oligonucleotide probes on microarray surface, and fluorimetric scan of array, processing of hybridization signal, and analysis of gene expression. The first phase is the Rna extraction from the biological sample. In this training, the starting biological samples is a frozen colon cancer samples, and the method used to extract the total RNA is based on adsorption to a silica gel matrix. The second phase, the preparation and labeling of target nucleic acid. The target nucleic acid is the molecule that we want to analyze in our procedure. In this case, we want to determine the levels of entire repertoire of cellular transcript and a large number of non-coding RNAs. The protocol requires the conversion of RNA in a form useful for the hybridization reaction. This conversion depends on the type of the microarray used. The procedure for Human Transcriptome Array 2.0 is summarized in the following slides. In this protocol, total RNA is first reverse transcribed to double cDNA. The cDNA is converted in antisense cRNA by in vitro transcription. Antisense cRNA is then reverse transcribed in a single-strand sense cDNA. Sense cDNA is then fragmented and labeled with biotin and represent the target cDNA ready to hybridize to antisense oligonucleotide probes on microarray surface. In this video, we will try to show the basic principle behind each of these steps, and we hope that the reason of such complex protocol will be clear at the end. Now, let's see the RNA extraction.
Biological samples are first homogenized in the presence of a highly denaturing guanidine thiocyanate containing buffer, which immediately inactivates RNases to ensure purification of intact RNA. In addition, lysate is passed through a 21-gauge needle attached to a sterile plastic syringe at least 10 times. Ethanol is added to provide appropriate binding conditions. The sample is then applied to a silica membrane mini-spin column. Total RNA binds to the membrane, and contaminants are efficiently washed away. After the washing steps, high-quality RNA is eluted in water. With this procedure, all RNA molecules longer than 200 nucleotides are purified.
The concentration of RNA should be determined by measuring the absorbance at 260 nanometer in a spectrophotometer. The ratio between the absorbance value of 260 and 280 nanometer gives an estimate of RNA purity. This ratio should be in the range of 1.7 to 2.1.
The first step of target preparation is the synthesis of the first strand of cDNA. 100 nanogram of RNA are used in this step. The reaction mixture is incubated in a thermocycler. During this step, RNA must be converted into cDNA in a reverse transcription, RT reaction using random primers containing the sequence of the T7 polymerase promoter. The reverse transcriptase synthesizes single-strand cDNA with the T promoter sequence at the 5' end. In the second step of target preparation, single-strand cDNA is converted to double-strand cDNA. The reaction uses RNase H. Renase H degrades RNA, and the DNA polymerase synthesizes the second strand of cDNA. In this step, the second strand of cDNA is the template, and T7 RNase polymerase binds to its promoter and synthesizes antisense RNA, also called complementary RNA or cRNA. This reaction is known as in vitro transcription, IVT. This step is also a form of amplification because many molecules of rcRNAs are synthesized of a single cDNA template. The reaction mixture is incubated for 16 hours at 40 degrees.
cRNA is purified by absorption of RNA of magnetic beads. The tube is moved to a magnetic rack and the beads are captured by the magnet. Supernatant is discarded and the beads are washed in order to remove enzyme, salts, inorganic phosphate and unincorporated nucleotides. cRNA is eluted by the beads using nuclease-free water.
So, let's see what happens in the lab. The tube is moved to a magnetic rack and the beads are captured by the magnet. When capture is complete, the mixture will be transparent. You can see the beads forming a pellet against the magnet. The supernatant is now being discarded. After washing in 80% ethanol, cRNA can be eluted in water.
"50 micrograms of cRNA is required for second cycle single-strand cDNA synthesis. In this step, cRNA is the template, and random primers and reverse transcriptase are used to synthesize sense-strand cDNA. However, the reaction mixture contains the OCUTP at a fixed ratio 1:5 relative to the OCTTP. Therefore, uracil will be present in the sequence of the sense-strand cDNA. In this step, RNase H hydrolyzes the cRNA template, leaving single-stranded cDNA.
After hydrolysis, the second cycle single-strand cDNA is purified by absorption to magnetic beads, as already shown in previous step. cDNA concentration is then measured by its absorbance at 260 nanometer. 50 microgram of input cRNA provide 5 to 15 microgram of single-strand cDNA. 5 microgram of purified single-strand cDNA are now required for fragmentation and labeling. As shown in previous step, the single-strand DNA contains the unnatural uracil base. In this step, uracil DNA glycosylase, UDG, specifically removes the uracil residue from the single-strand DNA molecules. In the same reaction, the apurinidinic endonuclease 1, APE1, cleaves the phosphodiesterase backbone at their basic site. The sites of DNA fragments are around 40 to 70 bases. The fragmented cDNA is labeled by terminal deoxynucleotidyl transferase, TDT, using the DNA labeling reagent that is covalently linked to biotin.
In this step, the fragmented and labeled target sense cDNA is added to the hybridization mixture. The hybridization mixture is incubated at 99 degrees Celsius for 5 minutes and then at 45 degrees Celsius for 5 minutes and is immediately loaded in the cartridge. The chip probe array consists of a square glass 1.28 cm for side mounted in a plastic cartridge. The array of oligoprobes is fixed on the inner glass surface. A plastic chamber is under the glass and acts as a reservoir where hybridization and washing occur. 2 holes in the back of the chip allow the loading of the chambers with solutions and its connection to the fluidic station. To load the chip with hybridization mix, I introduce the tip of a micropipette in one of these holes and the micropipette in the other one. After loading microarrays, they are incubated in a hybridization oven at 45 degrees with a rotation of 60 rpm for 16 hours. During incubation, sense target cDNA will hybridize to the corresponding antisense oligoprobes.
After 60 hours, the microarray are removed from the oven. the hybridization mix is removed from each array. Each array is filled completely with the wash buff array. The fluidic station is used to wash and stand the microarray, and it is controlled by a computer that increases the reproducibility of this technique. The chip is put into the cartridge holder, and in this way it is connected to the fluidic station by a tubing system. The fluidic station will wash the cartridge with two different washing solutions at a selected temperature and with decreasing ionic strength. The first washing will be at low stringency with the washing solution A. The second washing will be at high stringency with the washing B. The fluidic station will perform the staining step by 3 consecutive incubation with the solutions containing these tubes. First incubation will be with streptavidin and phycoerythrin. The second incubation will be with the biotechnology antibody against streptavidin. The third incubation will be with, again, streptavidin and phycoerythrin. The fluidic station will mix the staining solution by alternately draining and filling the cartridge. During hybridization, the sense cDNA hybridizes to antisense oligoprobe. In the staining step of the fluidic station, the streptavidin phycoerythrin binds to the biotin. A future step is performed in the fluidic station in order to amplify the signal. The array is incubated with a biotechnology antibody against streptavidin. A second incubation with streptavidin phycoerythrin completes the amplification step. Then the fluidic station fills the cartridge with holding buffer for scanning. The chip is loaded in the autoloader for the fluorimetric scan.
Fluorimetric scanner will provide fluorescence intensity value for each pixel of the macroarray. The software superimposes a grid on the image. The square of the grid corresponds to the probe cell on the macroarray. At this point, the software will convert the fluorescence intensity value per pixel in an average signal per probe cell, as we can see on the screen. The average intensity value per probe cells here are expressed in pseudo-color scale or in a gray scale. But we know that in a macroarray there are 6 million probe cells, and on average 100 probe cells correspond to a single transcript. Another dedicated software, will combine all the values of probe cell corresponding to a single transcript to provide 60,000 transcript values. The 60,000 transcript value represent the so-called gene expression profile per sample.
Finally, dedicated software are necessary to perform a comparison of gene expression between different samples in order to obtain a list of differentially expressed genes as splicing alternative events. The description of this analysis will require a different lesson. So, for the moment, we conclude at this point. Thank you very much for the attention.